I want you to picture something for a second. Close your eyes. Well, unless you're driving, in which case, please keep them firmly on the road. But visualize a simple wooden table.
Okay, I'm picturing it.
Sitting right there, in the middle of the wood grain, is a small pile of peanuts. You can see the rough, dimpled texture of the shells, maybe a tiny bit of dust settling around them.
It's a pretty common everyday image.
Exactly. And for a lot of people listening, that image is totally benign. I mean, it's a snack at a baseball game. Or it's the crunch in your favorite candy bar.
Right, but for a significant portion of the population, that innocent little pile of peanuts is essentially a biological landmine.
Yeah, a total landmine.
The anxiety that comes with that reality is just immense. It's relentless. You aren't just avoiding that visible pile of peanuts on the table. You're avoiding, say, a bakery that might have used the same cutting board 3 hours ago.
Oh, wow, right.
Or even a savory sauce that uses peanut protein as some unlisted proprietary thickener. It requires a constant, exhausting level of vigilance every single time you consume anything you didn't prepare yourself.
And that daily reality is exactly why we are diving into today's source material. We're looking at a deeply fascinating article published on May 16th, 2026 from technoscience.net.
It's a really incredible piece of research.
It outlines a major breakthrough coming out of McGill University. And the mission for our deep dive today is to explore how a piece of technology that, honestly, sounds like it was pulled straight out of a sci-fi movie, something called cold plasma, might actually be the key to permanently neutralizing these food allergies.
Yeah, cold plasma certainly evokes images of someone in a lab coat wielding a futuristic laser blaster.
Totally. It sounds like space-age weaponry.
It does, but the real genius of this McGill study is entirely grounded in the physical chemistry of how we can alter protein structures at a microscopic level. It's fundamentally about changing the physical 3D geometry of the food we eat rather than just trying to destroy it entirely.
So let's unpack the problem they're trying to solve first, because before we can appreciate how wild a cold plasma solution is, we really need to understand why the current standard methods for making peanuts safe are failing so badly.
Right, because the food industry hasn't just been sitting on its hands.
No, I mean, scientists have been trying to solve the peanut allergy problem for decades now.
They have. The global food industry has been desperately trying to crack this code because peanuts are just an incredibly valuable agricultural commodity.
Oh, for sure.
They're a massive source of plant protein, they're highly efficient to grow, they're calorie dense, and they offer this fantastic texture profile for manufacturing.
Yeah, the industry wants to use them in literally everything, from alternative milks to baked goods.
Exactly. The allergenicity is really the only major roadblock here.
So according to the article, the long-standing sort of go-to method for trying to reduce that allergic potential relies mostly on intense heat treatments.
Yes, thermal processing.
Right. Manufacturers essentially blast the peanut with thermal energy to try and destroy the allergenic proteins. But, okay, let's unpack this. Trying to remove a peanut allergy with extreme heat sounds to me like trying to disarm a bomb by using a flamethrower.
That is a brilliant analogy.
Like, sure, you disabled the bomb, but you also melted the entire house.
You disabled the threat, but you destroyed the very thing that made the food valuable in the first place.
Exactly.
When food manufacturers formulate a protein bar, or a non-dairy milk, or baking flour, they are relying on the protein behaving in a very predictable way. We call these functional properties.
Functional properties, gotcha.
Right. A protein needs to be able to bind with water correctly. It needs to emulsify fats. And, it needs to maintain a neutral or pleasant taste profile.
And I'm guessing a flamethrower doesn't help with that.
Not at all. Applying that level of intense thermal stress denatures the protein so severely that those functional properties are completely obliterated.
So it's not really a peanut anymore. It's just a burnt, useless husk of protein that won't blend into a smoothie or bake into a cookie.
Precisely. That renders it totally useless for consumer products. Because heat destroys the food's utility, food scientists have spent years exploring non-thermal approaches.
Which makes sense.
And the most prominent alternative has been irradiation. That's exposing the food to ionizing radiation, like gamma rays or X-rays, to try and break down the allergenic proteins without cooking the food.
Okay, so in theory, irradiation avoids the melting the house problem. You aren't using a thermal flamethrower. But the source material notes that irradiation has yielded incredibly frustrating and variable results in practice.
It has. And the mechanism behind irradiation explains that inconsistency. When ionizing radiation hits a complex food matrix like a peanut, it acts like a shotgun blast.
A shotgun blast.
Yeah, the radiation waves split water molecules inside the peanut, creating highly reactive free radicals that then just bounce around and damage the proteins.
Oh, I see.
The problem is that peanuts are agricultural products. One batch might have been harvested in a slightly wetter climate and has a higher moisture content, meaning the radiation creates more free radicals.
Okay, and then the next batch.
The next batch might be drier or have a slightly different fat composition, which shields the proteins differently.
So you might process one batch of peanuts and see a 20% drop in allergens and process the next batch the exact same way and see almost no change at all.
Exactly. And when you are dealing with a severe, potentially life-threatening anaphylactic allergy, a shotgun approach that relies on the natural moisture variations of a crop is entirely unacceptable.
Yeah, you can't gamble with that.
You really can't. The inconsistency makes it completely unscalable for broad commercial safety.
Since heat destroys the food and irradiation is a dangerous gamble, researchers were basically forced to look outside traditional food science entirely to find a new physical mechanism.
Which brings us to the core of the breakthrough.
Right. The research was led by Jin Wang, who did this work as a PhD student at McGill and is now a professor at Southeast China University, along with his co-author Vijaya Raghavan.
And they decided to utilize cold plasma.
Okay, so to understand their approach, we probably need to define cold plasma.
We do. We are all familiar with the 3 standard states of matter, right? Solids, liquids, and gases.
Right, basic science class stuff.
Exactly. Well, plasma is widely considered the 4th state of matter. It is a gas that has been energized, usually via an electrical field, to the point where electrons are actually stripped away from their atoms.
Oh, wow. So what does that leave you with?
What you are left with is an ion-charged, highly reactive gas. Because it is so energetically charged, it forces rapid chemical changes in the environment and the materials it comes into contact with, completely without relying on extreme heat.
So is cold plasma basically like a microscopic, localized lightning storm in a lab? Like you are harnessing this controlled electrical storm to subtly reorganize the peanut's molecular furniture without starting a fire?
That's a perfect way to visualize it. It is a controlled storm that interacts directly with the surface and the structure of the molecules it surrounds.
Okay, so they have this tiny lightning storm.
Right. But what's fascinating here is not just the tool itself, but the specific methodology they applied to it. In typical food science research, when someone wants to test a new technology on an allergen, they will isolate the one specific allergy-causing protein.
They just pull the bad protein out?
Yep, they put it in a pristine lab environment like a petri dish and they zap it. But this McGill team deliberately chose not to do that.
Why not?
They extracted the whole peanut protein matrix and treated that entire complex mixture with the cold plasma.
But wait, isolating the single bad protein seems like it would yield much cleaner data.
Right.
Why go through the trouble of testing the entire messy matrix of the peanut?
Well, Vijaya Raghavan points out a critical flaw in relying solely on isolated protein testing. In the real world, consumers are never exposed to a perfectly isolated lab allergen.
Oh, right, because you're just eating food.
Exactly. When you eat a peanut butter cookie, your immune system is encountering a massive tangled web of different proteins, starches, and fats all interacting with each other.
It's a whole package deal.
It really is. Proteins in a real food matrix actually form physical structures together. Some proteins act like molecular chaperones, folding around and shielding other proteins.
I get it now.
Yeah.
If you only zap the isolated protein in a petri dish, you have no idea if the surrounding fats and proteins in a real peanut would have shielded the allergen from the cold plasma storm.
Exactly. Testing the isolated protein gives you a false sense of security. By extracting the whole protein and evaluating it in vitro, meaning in laboratory tests outside of a living organism, they were forcing the technology to prove itself in a highly realistic, challenging scenario.
They really put it to the test.
They had to. They needed to know if the plasma could penetrate the complex entanglement of a real food product and still fundamentally alter the allergenicity.
Okay, so they exposed this tangled, whole peanut protein matrix to the cold plasma storm. To measure the success of the experiment, the researchers evaluated what they call the protein's immunoreactivity.
Yes. Immunoreactivity is the core metric for food allergies. We often hear allergies described as a lock-and-key mechanism, but that implies something rigid and metal.
Right.
When dealing with complex biological proteins, a better way to visualize immunoreactivity is to think of the allergen as a flat piece of paper that has been intricately folded into a highly specific origami swan.
Origami swan.
Your immune system's antibodies are trained to constantly patrol your bloodstream looking for the exact three-dimensional shape of that swan. If it spots the swan, it binds to it, sounds the alarm, and triggers a severe allergic cascade.
So the immunoreactivity is just a laboratory measurement of how easily the immune system recognizes that specific origami shape.
Yes.
So the goal of the cold plasma isn't to obliterate the paper, because that would ruin the food, but rather to act like a sudden gust of wind that unfolds the swan and crumples it into an unrecognizable ball.
That is the precise physical goal. It is the exact same piece of paper, the identical sequence of amino acids, but because the three-dimensional geometry is no longer shaped like a swan, the immune system's antibodies simply float right past it.
Because they don't recognize it as a threat.
Exactly.
So the microscopic lightning storm actually managed to crumple the origami swan.
The results were staggering. After just 25 minutes of exposing the whole peanut protein to the cold plasma treatment, the team observed a near 70% reduction in overall immunoreactivity.
Wait, really? Nearly 3/4 of the allergic potential was completely neutralized in under half an hour.
Yes. The shape of the protein was fundamentally changed.
That is wild.
The immune system's ability to bind to the protein was drastically severed.
Here's where it gets really interesting, though. Because as I was reading through the source material, I noticed they didn't just neutralize the allergy threat.
Right, there was more to it.
The cold plasma treatment triggered this massive secondary benefit. The study showed that treating the peanut this way actually made it significantly easier for the human gut to break down and digest the protein.
It did. And one of the consistent challenges with plant-based proteins is that they can be difficult for the human digestive system to process efficiently.
Because they're tough to break down.
Right, the proteins are tightly wound up. But as the cold plasma was busy disrupting the structure of the allergen, crumpling the origami swan, it was simultaneously unraveling the broader protein structure.
Oh, so it loosens everything up.
Exactly. That unraveling process exposes far more surface area. When the protein is opened up, our natural digestive enzymes have much easier access to latch onto the protein chains and break them down for cellular absorption.
So it made the food safer and nutritionally superior.
A rare double win in food science.
Okay, a 70% drop in immunoreactivity is incredible, but practically speaking, a 30% remaining allergic potential is still a massive biological landmine for someone with a severe anaphylactic allergy.
That's a very important point.
Does a 70% reduction mean I can safely eat this if I have a severe allergy, or is it just the foundation for making an allergy-friendly protein powder? Because I can't imagine this means someone with a severe allergy can just grab a handful of whole, plasma-zapped peanuts at a bar.
If we connect this to the bigger picture, eating whole peanuts is not the immediate goal of this technology. Raghavan's vision, as outlined in the research, is strategically focused on manufacturing.
Oh, manufacturing, okay.
Right. A 70% reduction paves the way to create highly functional, hypoallergenic peanut protein powders.
So they use the cold plasma to create a safe, stable powder that manufacturers can confidently swap into their processed foods instead of using dangerous, untreated peanut derivatives.
Exactly. It allows massive food companies to safely utilize the nutritional power and the functional properties of peanut protein without putting a portion of their consumer base at risk.
Which is huge for the industry.
It is. This opens the door for entirely new product lines of plant-based milks, baking mixes, and protein bars that carry a significantly reduced risk profile. It is a targeted strategy to secure the food supply chain and eliminate the pervasive anxiety of manufacturing cross-contamination.
Okay, so we have established that this microscopic lightning storm effectively crumples the origami shape of the protein, hiding it from the immune system while making it easier to digest.
Yes.
But to really shortcut our way to being well-informed on this topic, we need to understand the underlying chemical mechanism. Like, how is a cloud of charged gas physically forcing a protein to unfold?
It's all about the chemistry.
The article highlights one very specific chemical player driving this 70% drop.
This is where the McGill study makes a significant departure from previous scientific assumptions. Prior research into applying cold plasma to foods broadly suggested that the chemical changes were being driven by a combination of generated compounds, specifically hydrogen peroxide and nitrates.
Okay, so a mix of the two.
Right. It was assumed both were acting as the active agents breaking down the proteins.
But when Wang and Raghavan tested the whole peanut protein matrix, they didn't see hydrogen peroxide playing a major role at all. The study found only an increase in nitrite levels.
Yes.
And Raghavan explicitly notes that nitrites play the preeminent role in modifying the protein structure here. But let me push back on this for a second.
Sure.
Because when I hear the word nitrites, I immediately think of the preservatives in hot dogs and processed deli meats that every health blog tells us to strictly avoid. How are they acting as the heroes here?
The confusion is incredibly common, and it is crucial to draw a sharp line between dietary nitrate salts and plasma-generated nitrites.
Okay, so they're different.
Very different. When we talk about avoiding nitrites in cured meats, we're talking about physical salts that are dumped into meat vats to preserve a pink color and prevent bacterial growth.
Yep.
Over time, in high quantities, consuming those salts can lead to the formation of harmful compounds in the body. But in the context of cold plasma, nobody is dumping a bucket of preservative salt onto a peanut.
If they aren't adding a chemical powder, where are the nitrites coming from?
The cold plasma is literally synthesizing them out of thin air.
Out of the air.
Really? Yes. The ambient air around us is roughly 78% nitrogen and 21% oxygen. Normally, nitrogen gas is incredibly stable. It doesn't want to react with anything.
Right, it's just inert.
But when you introduce the intense, localized electrical energy of the cold plasma, it rips those stable nitrogen and oxygen molecules apart.
Oh, wow.
The atoms are forced to recombine on the surface of the peanut into reactive nitrogen species, which includes these microscopic, localized nitrites.
So the energetic storm manufactures its own chemical tools directly from the air surrounding the food.
Exactly.
Once those nitrites are synthesized on the surface of the peanut, how do they actually crumple the origami swan?
They act as incredibly precise molecular scalpels. The 3D origami shape of a protein is held together by structural pillars, most notably strong connections called disulfide bonds.
Disulfide bonds.
Think of disulfide bonds as the rigid steel girders holding up a building. When the plasma generates these nitrites, the nitrites specifically seek out and interact with certain amino acids like cysteine and tyrosine that form those girders.
Okay, so they target the structural supports.
Right. The nitrites attach themselves to the sulfur atoms, chemically severing the disulfide bonds.
So you basically cut the steel girders, and the entire structure collapses permanently.
The structural integrity fails, and the protein folds in on itself into a new, unrecognizable shape. Because the immune system relies entirely on recognizing the specific intact architecture of the original shape, this nitrite-driven collapse permanently hides the protein from the body's alarm systems.
That is so elegant.
It is. And Raghavan points out that this precise chemical mechanism, the synthesis of nitrites and their specific severing of amino acid bonds, is frequently overlooked in applied research. Food scientists often just note that cold plasma works and move on, without ever fully interrogating the chemical pathway driving the change.
But understanding that specific mechanism changes everything about how you deploy the technology. If you know that the synthesized nitrites are the actual scalpels cutting the girders, you can engineer the cold plasma chamber to optimize the exact mix of nitrogen and oxygen needed to maximize nitrite production.
Exactly, without wasting electrical energy or causing unwanted side effects to the rest of the peanut matrix.
That level of understanding is what allows you to refine a lab experiment into an industrial scale reality.
It's the key to scaling the whole process.
So what does this all mean for you, the listener? We have explored a highly optimized, non-thermal technology that disarms one of the most volatile allergens on the planet. But the implications of understanding this nitrite-driven protein collapse stretch far beyond a simple peanut allergy.
They really do. Raghavan and his team at McGill are absolutely not stopping at the peanut.
I would hope not.
Now that they have proven the efficacy of testing the whole protein matrix and identified the crucial role of localized nitrates in severing those structural bonds, they are planning to optimize this exact technology for a wide array of problematic food allergens.
Oh, that's exciting. Like what?
The research specifically highlights ongoing plans for eggs and hazelnuts.
Hazelnuts. If you can deploy cold plasma on hazelnuts, the global chocolate spread industry is going to experience a massive shift.
Absolutely.
But eggs seem like the truly universal application. I mean, egg proteins are ubiquitous across almost all food manufacturing, baking, pastas, sauces, binders.
They're everywhere.
If this team can create a highly functional hypoallergenic egg protein powder using cold plasma, they fundamentally change the safety landscape of the modern supermarket. You completely remove the lingering threat of manufacturing cross-contamination.
It would be a monumental shift in food safety.
So let's briefly recap the journey we've taken through the source material today. We started with an industry heavily reliant on thermal flamethrowers that practically ruined the functional properties of the food they were desperately trying to make safe.
Right.
Then, Wang and Raghavan step in with cold plasma, a microscopic lightning storm. Instead of taking the easy route of zapping isolated lab samples, they rigorously tested the complex, entangled, whole peanut protein matrix.
Which was the crucial decision.
We learned that the plasma energizes ambient air to synthesize localized nitrites, which act as molecular scalpels to sever the structural girders of the protein, permanently crumpling its 3D shape so the immune system just ignores it.
The result is a staggering 70% drop in immunoreactivity in just 25 minutes, entirely avoiding the destructive nature of heat.
And doing double duty.
Yes, simultaneously unraveling the protein to make it far easier for our natural digestive enzymes to process. It is a masterclass in revisiting an old problem with a new technological lens and a willingness to understand the deep chemistry at play.
It really is a remarkable scientific pivot. And it leaves me with one final thought to ponder. We spent this deep dive exploring how cold plasma and synthesized nitrites can successfully crumple and refold dangerous proteins so they bypass our immune system's alarm bells.
Right.
But we also clearly noted this secondary benefit, that the exact same unraveling process made the peanut protein significantly easier to digest and absorb.
It's a huge bonus.
So here's the question I want to leave you with today. If this non-thermal technology can be fine-tuned to intentionally sculpt and re-engineer the physical shape of a protein matrix, could it eventually be used not just to hide the bad stuff, like allergens, but to actively optimize the good stuff?
Oh, that is a fascinating thought.
Right. Could we be looking at a near future where cold plasma is routinely used to pre-fold and unravel the proteins in our everyday non-allergenic foods, specifically engineered so that our bodies can absorb the daily nutrition twice as fast with zero digestive strain?
It's definitely plausible based on this research.
The next time you sit down at a wooden table and see a simple pile of peanuts, don't just see a baseball snack, and don't just see a biological landmine. See a complex microscopic puzzle that we are finally figuring out how to solve and reassemble, one molecular fold at a time.